So welcome back, and I will be talking about in-gel gymography. Now there are two terminologies here. One is called as in-gel and one is called as gymo, and one is graphy. Like three means gymography, maybe one word. So in-gel, which suggests that it is within a gel which is working. The gymography is a kind of a graph which you are getting for gymogens. Okay, so gymogens. Now the gymogens are certain enzymes which basically are in an inactive form, and then they have to be activated. Like your metalloproteases is one of the classes, and it is used for several purposes, but we will discuss about like the gelatin in gel gymography. So you are looking for a gelatinase, and as well as some casein degrading enzymes. So let's look into this. So the definition, as you can say, is an enzyme assay based on SDS-PAGE that evaluates a protease ability to digest a substrate. Now there is a variation to this also. The variation to here is that in SDS-PAGE, if you remember in the previous video, we talk about that this usually use a reducing and a non-reducing condition, and in a reducing condition even you boil your samples before loading to make it linear. In non-reducing, and you also add your reducing agent like BME, that is beta mercaptoethanol, or DTT can be added. In non-reducing condition you don't boil them and you don't add your reducing agent. So if there is a subunits, then they will be separated out. The other variation which is in gel gymography is, let me draw here. So let's say in a typical gel you have these wells, right? These are your wells. This is your stacking gel, and this is your resolving, make it separating or resolving gel. Now in case of this, you cast a whole gel like this, means there is no pH variation or so. So this is whole resolving gel, and then there are wells like this. Other way which you can do here is, this is both is your SDS-PAGE. So this is your SDS-PAGE, this is your SDS-PAGE. The other thing, there is a variation which one can do here, is related with your native page as well. Means you need not to use any SDS. So that is also can be done for in gel gymography. So let me rub this. So on a running a gel with a substrate copolymerized with acrylamide. So there are, again, when you are making these gels, means when you are making this resolving gel or one gel in a go, you basically add your substrate. Okay, whatever the substrate, as I said, you can add your gelatin, or you can use casein, you can use collagen, whatever, so many heparin, so others are also used. So that is one way, that is you when you are pouring your gel, then you can add. And the second way which can be done is but then that don't fall into the preview of in gel, that people can run the gel because the proteins are there, then you can put in your substrate solutions as well. Okay, but that is a different chemistry that is used, that I maybe tell after the end that how that can be done. But right now we will focus in gel gymography, where you are adding your substrate while preparing your gel. Now, as I said, we will be focusing here only on two parts, that is the gelatin gymography and casein gymography. The resolution of casein gymography is not very good, and that's why some people have to run it twice a gel. The reason for that is that caseins are small molecules, roughly around 22 kilodalton. And if you remember or recall from your SDS-PAGE, that is the small molecules, they are in the lower side. So that's why in casein gymography, what is happening, they are also slightly charged. So when you are running your gel, they are not like a neutral compound. Because of the slight charge, they also migrate. Okay, so they also try to migrate in this direction. So they will, more of the caseins will be at this end, at the end, and your whole protein is here, then that could be an aromatic. Okay. Then the other one is your gelatin gymography. In the gelatin gymography, you use gelatin while preparing. So let's look into, maybe in 2-3 minutes I will wind up here, because there's not much here required. So gelatinase, which is used here as an example, this is the metalloproteases. Now what are the details? You can refer to a paper. There is a link to this paper which is in this DOI, you can refer to. Okay. And this is as you can see, this is a in gel gymography. What they are trying to show, there are certain discrete bands. Okay, there are certain discrete bands which is shown here. Now this discrete bands is different classes of your matrix metalloproteases complexes, and you can able to find out whether there is, you can create some mutations and then you can see that is working or not, or you can also differentiate between a proteases with the other one. The second one here is, this is your casein gymography. Okay, this is also taken from a different paper. In this casein gymography, they have shown here 3 lanes. Okay, 3 lanes. This is your protein marker lane, this is your whole protein in a gel, and this is shown here different, the same, like they also use a matrix metalloproteases. Matrix metalloproteases are also actually involved in cell signaling and the growth and development. So you can see clear bands like this. Okay, this is the bands as you can see, and you can differentiate in the gel. So the advantage here, as I said, different isoforms can be found out. You can differentiate between a zymogen with another zymogen, and the difference here is that you use a non-reducing condition. Okay, so in case of like how you do this process, so in a very briefly, because more or less it has been mentioned in other videos also, like and this is specifically mentioning about the casein gymography, like you do not heat your samples. Okay, because you do not want your protein to be degraded, because these are enzymes. So you run the gel was submerged in the casein. So that is the other way that you, after your electrophoresis process is done, you can submerge. So I was talking about the other way, that how this experiment can be done. Then you incubate that with at 70 degrees Celsius for 10 minutes, so that it can work upon. And why that 70 degree? Again, it depends upon the proteins or the zymogens which you are working. They may be different, so that's why you use maybe this is heat, that's why you use. And I will tell, as I said, my personal story as well. Then you stain the gel with Comasu Brilliant Blue, and then you destain your gels. So staining and destaining process, and then you can see the clear blue background bands. So this is a paper they are referring, and they actually use different things which you can make it out here. Like for example, the control, they have not added anything, but when they use the protease inhibitor, okay, that is the PMSF, phenyl methane sulfonate fluoride. So then when they use this, see, the activity has decreased. Why? The reason being because this is a protease inhibitor. So if there is any protease activity which is there for casein, it is not it will hamper it. So that's why your activity shows decrease, means your bands will look faint or the size will be different. When they use this with the EDTA, that is the chelating agents, so but increasing the concentration, they see there is activity reduced, and then they checked for certain other conditions. Okay, so this work for some or not, this you can refer to that paper, which link as I'm saying here, this one, and you can refer to it. I will share that link as well. Now there is one more thing which is required to be told here, that is the denaturation and renaturation step. Okay, now this step is required for certain kinds of like you basically don't use a non-ionic detergent instead of using a ionic, because SDS is also a ionic detergent. So some of the proteins, they may not work on that SDS. So because if you are running angel gymography and you're not getting anything, then you can restore the activity. Okay, and how can you restore the activity? Maybe say first you have run the SDS page, okay, like with the your co-substrate. Your co-substrate means whatever the substrate, and with the acrylamide you run the gel. Before that, what, and your protein, let's say, gets denatured here. So to restore the activity with the same gel, okay, what you can do is that you can put in a non-ionic detergent like Triton X100, which exchange with your SDS detergent. So it is like a dialysis which is happening, and based on those dialysis, then you incubate your gel at 37 degrees for overnight with the buffer and as well as the cofactors, like if required calciums, magnesiums, and then your enzymes will be enough for your activity. So if you are not finding anything in your gel, don't get disheartened. Maybe that SDS is not working properly, or it is denatured your protein. Then you can use this as a another method. So before I end, let me summarize what I said. So we actually talk about the SDS based in gel gymography. Now in this particular case, the difference here is that you don't you non-reducing condition, no heat is used. Second thing which I mentioned, that it has to be with your substrate. Okay, whatever the substrate you add in the gel itself, so that you can see what type of activity is there. I showed you examples of both the casein and the gelatin gymography. The third thing which I mentioned here is that if you are not getting your activity, then in that particular case you can renature your protein, and that renaturation steps can be done because of the Triton X, because some of your proteins might be denatured in the presence of the SDH. So that possibility is there. The fourth point which I also mentioned, like which some people do, that they are not going with the in gel means they are not using the substrates in gel, but rather they are incubating the gel after your proteins has been resoluted with a particular substrate, and then they see that there what is the bands there or not. So there are advantages with this method is that you can find out, as I said earlier also, that different isoforms or zymogens can be differentiated. The disadvantage with this method also, because we have to look into the limitations, that some of this is little bit cumbersome process. The second thing is that you cannot find out the exact activity. Okay, so you cannot quantify, you can semi-quantitatively quantify, but not the exact quantity can be measured with in gel gymography. So with this I will stop here. So thank you.